participants. Thank you very much for registering for this month's JETZON Open Call. We have two excellent talks, as you will have seen from the advertisements. And so to get started, I would like to introduce Nick Bock from Columbia, who's going to talk to us about biogeographical classification using BGC-Argo floats. Nick, over to you with thanks.
Cool. Thanks so much, Adrian. Thanks so much to everyone for joining. Let me take 1 second to share my screen here. Switch over. We'll drag. All right. So again, hello. My name is Nicholas Bock. I'm a postdoctoral researcher at Columbia University Lamont Doherty Earth Observatory. And the title of this presentation is Biogeographical Classification of the Global Ocean from BGC-Argo Floats. A lot of what I'm going to talk about today is based on recent results of a recent manuscript of the same title, actually, which was completed in collaboration with Drs. Marin Cornick at University of Washington School of Oceanography, Dr. Hervé Klost at Villafranche Oceanography Laboratory, and Dr. Solange Duhamel at the University of Arizona. And the QR code on the right here links to the paper for easy reference if you want to have a look. So just to give some broad context for what I'm going to talk about, there is, of course, a huge amount of spatio-temporal variability in in biological processes within the global ocean, as captured here by this time lapse of chlorophyll A concentration measured by MODIS during 2021. And so understanding or modeling these processes in part, of course, requires identifying regions where this variability might be expected to be relatively constrained. Just 1 second, sorry. Let me drag the chat window out of the way here. Sorry about that. And so that's the overall goal of biogeographical classification. And so many classification methods have been devised over the last 80 years or so. One of the most famous ones is the Longhurst provinces, which used a combination of data from global climatologies and remote sensing platforms to define the 56 ecological provinces captured here. Other classifications have relied on ocean color data alone. For example, the classifications used by Behrenfeld et al. 2005, which is just based on long-term variance in chlorophyll A concentrations based on satellite data. The different variance bins were then used to guide parameter selection for modeling parameter production. In all these cases, though, there's been really limited opportunity to account for subsurface variability in whatever the classification criteria selected are, just because satellite images primarily capture what's happening at the surface, and ship-based measurements provide pretty limited, fairly limited spatio-temporal resolution. And so the BGC-Argo network happily addresses both of these constraints to some extent, providing tens of thousands of profiles of biologically relevant parameters across the global ocean, and a potential basis for biogeographical classification that takes all this subsurface variability into account. So over the next couple of slides, I'm going to talk about a method that we devised for using singular value decomposition to determine biome membership for a large data set of biooptical time series from BGC-Argo floats. Then I'm going to talk about some of the seasonal trends in phytoplankton distributions and their associated environmental drivers that we identified for each of these biomes, along with some of the seasonal trends in mesopelagic particle concentrations for each biome. Then I'll close with a quick mention of some further applications for this method and some next steps for the project. So with that, I'm going to take a sharp detour to talk for a minute about eigenfaces. And the eigenfaces concept is really widely used as an example of how singular value decomposition or principal component analysis works, and is really analogous to our approach for classifying float data. So the idea is that you take some collection of photos, you convert all this image data into a matrix, you perform singular value decomposition on this great big matrix of image data, and then you get this series of eigenfaces, each capturing different components of the variability in the original data set. Aside from these eigenfaces, the SVD results also provide you with a set of coefficients specific to each of the original images that can be used in concert with the eigenfaces to reconstruct the original data. So you take 3 of eigenface 0, 2 of eigenface 1, 1 of eigenface 2, so on and so on and so on, and you get this image of Dwayne Johnson. You take a separate set of coefficients and you get LeBron James or Queen Elizabeth. And so you don't just get these eigenfaces, but you get these coefficients that really effectively summarize the different sources of variability contributing to the original images. And so this is more or less what we did with the float data, except instead of using photos, we used interpolated time series of chlorophyll A fluorescence and optical backscatter from 422 or across 422 annual time series with a subset being pictured here. And so compiling all of this data into a great big matrix and performing SVD on that, we obtained this set of eigen time series for chlorophyll A on the left-hand side and optical backscatter on the right-hand side. Of course, we also obtained this set of time series specific coefficients. And by performing cluster analysis on the first 6 of these coefficients, which reflected something like 83% of the total variability in the original data set, we were then able to objectively classify these 422 original time series as belonging to one of 6 biomes. So that included the equatorial biome here, the Arabian Sea Biome, the permanent deep chlorophyll maximum biome, which consisted of time series concentrated in the central gyres of each basin, the seasonal DCM biome, which consisted of time series in the Mediterranean Sea and the subtropical frontal zone, and the low chlorophyll bloom and high chlorophyll bloom biomes, which consisted of time series in the Southern Ocean and the North Atlantic, respectively. And so this map on the left shows the global distribution of time series belonging to each of these biomes, with each point corresponding to the median latitude and longitude for one of those 422 annual time series. And the plot on the right shows the latitudinal distribution, really highlighting that the biomes were distributed more or less symmetrically about the equator. Okay, so because each one of these points represents a full year's worth of profiles, this approach provides a huge amount of data to evaluate ecological processes, and they're within each one of these biomes. So, for example, by calculating the average of the chlorophyll A fluorescence time series, it's possible to get a clear picture of seasonality in phytoplankton distributions and physiology for each biome. From these results, it's clear that the equatorial and the seasonal deep chlorophyll maximum and the permanent deep chlorophyll maximum biomes are characterized by this really prominent deep chlorophyll maximum of increasing depth from the equatorial to the permanent DCM biome. While the low chlorophyll bloom and high chlorophyll bloom biomes are characterized by, as their names suggest, these really large summer phytoplankton blooms, with an especially high contrast in chlorophyll A fluorescence between winter and summer months in the high chlorophyll bloom biome. And finally, it's a little bit difficult to tell from this image, but the Arabian Sea biome is characterized by this bimodal seasonality in chlorophyll A fluorescence due to the influence of the Asian monsoon, with the formation of a deep chlorophyll maximum during intermonsoon periods and the formation of phytoplankton bloom during the monsoon periods. Okay, so we can do the same thing with our time series of optical backscatter measurements, which provides a really synoptic view of particle distributions within each biome. And the particle concentrations were more or less well correlated with chlorophyll A fluorescence values, with a couple of fairly conspicuous exceptions. So, for instance, this really prominent backscatter maximum band in the Mesopelagic zone in the Arabian Sea Biome, the relatively homogeneous distributions of backscatter values or particle concentrations in above the deep chlorophyll maximum in the equatorial and permanent DCM and seasonal DCM biomes, presumably reflecting heterotrophic bacteria or weakly fluorescent phytoplankton cells. And then finally, even though in preparing our data, we removed spikes from the backscatter signal, which might correspond to larger particles, that is, particles larger than 20 microns in diameter. Our results nonetheless capture the elevated concentrations of large particles like these ones in the mesopelagic zone in high latitudes, especially in the high chlorophyll bloom biome, as reflected by all this splotchiness in the mesopelagic zone here. Okay, so but beyond using the bio-optical measurements to get a sense of phytoplankton distributions, we were also able to characterize the environmental context of each biome using float measurements of physical and biogeochemical parameters. So, for example, we used temperature and salinity data to determine the average mixed layer depth for each biome, as shown by the black line here. We used measured and estimated nitrate concentrations, shown by the red, to estimate the depth and the slope of the nitrocline, with the nitrocline depth being shown by the red line here. And just for clarification, we defined the nitrocline depth as the depth where nitrate first reached concentrations 1 micromolar greater than those at the surface. And then finally, we use data from float radiometers to estimate daily integrated PAR, both within the average daily integrated PAR within the mixed layer and at the depth of the nitrocline itself. For the sake of convenience, this is indirectly illustrated by the depth of the 0.1% PAR isolume, shown in yellow here. And so a few takeaways from these results were, first, that the equatorial and Arabian Sea biomes were characterized by particularly high light levels at the depth of the nitrocline, as reflected by the 0.1% PAR isolume being located well beneath the nitrocline depth across almost the entire year in either case. While in the permanent DCM and seasonal DCM biomes, the nitrocline depth was more or less coincident with the 0.1% PAR isolume. By contrast, the low chlorophyll bloom and the high chlorophyll bloom biomes were primarily differentiated from the others based on the depth or the extent of the seasonal variability in the mixed layer depth, as shown here. Okay, so with that sort of picture of what was happening, what we found in the photic zone for each biome, I want to pivot to talk for a minute about what our results told us about particle stocks in the mesopelagic zone. So the float, the BGC-Argo floats do complete a transit between 1,000 meters beneath the surface and the surface. So we have measurements not just from 0 to 500 meters, like I was showing on the previous slides, but all the way down to 1,000 meters. And so in our analysis, to evaluate what was happening in the mesopelagic zone, we defined the mesopelagic zone as the range of depths between the 0.1% PAR isolume and 1,000 meters beneath the surface, as indicated by this bracket here. And then we defined the productive zone as the area or the range of depths above 0.1% PAR isolume, as shown by this smaller bracket here. And so comparing the average backscatter values within the productive zone, shown on the x-axis here, to a proxy for the transfer efficiency, defined as the average backscatter within the mesopelagic zone over the average backscatter within the productive zone, and shown on the y-axis here, we were able to identify this global inverse relationship between euphotic zone particle stocks or productive zone particle stocks and transfer efficiency. We were also able to determine that this trend, this global trend, was primarily driven by extreme values in high latitudes during winter and summer months, respectively. To get a clearer sense of how particle attenuation itself contributed to this overall trend, we then subdivided the mesopelagic zone into 4 equally sized layers, as shown here on this example profile of backscatter values, and then calculated the average backscatter value within each depth range. So then we were able to compare the average particle stocks within the productive zone, shown on the red, shown by this red line here, to the average particle stocks within each one of these layers, shown in light to dark gray, with light gray being nearest the surface and dark gray or black being furthest from the surface or closest to the bottom of the mesopelagic zone. And the distance between these lines corresponds to the instantaneous particle attenuation with depth. And these results really highlight that the productive zone particle stocks, especially in the seasonal DCM and the low chlorophyll bloom and the high chlorophyll bloom biomes, are really directly related to particle attenuation with depth, as might be expected based on the results on the previous slide. With that, just to wrap up, I want to talk a little bit about further applications for the method and some next steps, what we'll be working on next. So in particular, I want to highlight the recent work of Elsa Simon, who is a PhD student at Villafranche Oceanography Laboratory, and who used this SVD classification approach to evaluate the functioning of the biological carbon pump across 5 high latitude biomes captured on this map here. And so Elsa was really able to expand on the analysis of the Mesopelagic zone particle stocks that I was just discussing a few slides ago, in particular, estimating particular organic carbon based on the backscatter values, and then looking at rates of seasonal accumulation in POC for each one of those biomes, as illustrated by the slope of the green lines on this plot of seasonal trends in backscatter concentrations, or sorry, POC concentrations. She was then able to look at fluxes for each biome based on by fitting a power curve to the slope of this line for each one of these layers, as shown in this plot on the lower right-hand side. By repeating this process for oxygen data, she was also able to compare how the efficiency and intensity of the biological carbon pump related to respiration rates. That'll be exciting to see as she continues working on it. In all these results, though, the biomes remain defined as discrete time series. And so sort of the big next step, for me at least, is to explore methods for extrapolating these classifications to areas that are not directly sampled by floats. So in other words, is it possible to give a machine learning classifier a set of geographical coordinates and remote sensing measurements for a set of classified profiles, and then have that classifier predict biomembership for data that it hasn't seen before, for instance, from a satellite image. And so sort of this is the first effort at this. I took 26,000 classified profiles with satellite matchups from our original data set, used them to train a random forests classifier, which then was able to predict biomembership for new data in the test data set with an error rate of approximately 2.5%, as highlighted by this confusion matrix, which just sort of shows the proportion of predictions that were correct or incorrect. So applying this model to a global data set of the classifier predictors, it's then possible to extrapolate the original SVD classifications to the global scale, as shown here. And so while there is sort of some wonky stuff happening at low latitudes here and there, a refined version of this approach would address or achieve the larger goal of creating a global biogeographical classification based on subsurface measurements from BGC-Argo floats, as outlined at the start of the talk. But that's what I have. I'd like to really quickly thank my collaborators again on the project, in addition to support from the National Science Foundation and the ERC-funded REFINE project, and of course, everyone on the BGC-Argo data management team. I look forward to whatever questions you might have. And of course, please don't hesitate to reach out as well. My email address is right here. So thanks again.
Excellent. Thanks, Nick. Lots there to discuss, I think. So let me just see if there's anything yet I haven't got anything yet in the chat as far as I can tell. Let me just have a quick look. So if anybody, so Nick Casso, do you want to ask a question? I see you've got your hand up.
Hi, thanks, Nicholas. This was great. A quick question about the number of biomes. How do you decide on the number of biomes? Is that decided by the SVD, or do you decide how granular the biomes are?
Yeah, so that was based on the results of the clustering algorithm. So 6 really provided, we evaluated the quality of the clusters based on the silhouette score. That's sort of like the average distance of each time series to the biome that it's assigned to versus all the other biomes. And 6 clusters or 6 biomes give us the highest possible silhouette score, or the highest quality clusters, in other words.
Thank you.
Thanks. Ken, I see you've got a question.
Yeah, very cool stuff. I love the concept and what you've done. But when you start using backscatter and calling it POC flux or flux, that there's a lot of steps there that aren't necessarily captured. Would you agree with that, or do you think if you just measure backscatter, you can predict carbon flux in the ocean? What are some comments on that?
Yeah, so I would say that our results as far as backscatter is concerned really, only captures what's going on at the sort of highest level. There's of course a lot of nuance whether it all in fact reflects POC versus other suspended particles, etc. Whether that relationship is consistent with depth, etc. And so I would agree with you that there is a lot of nuance to be considered in trying to evaluate exactly what the backscatter represents. And that was part of why I sort of tried to limit the conversation just to looking at the backscatter values itself without extrapolating to or trying to identify conversion coefficients, which would be interesting to look at, but sort of beyond the scope of what we are trying to work on.
And it might be fun if you have the stock of POC and the seasonality, you can look at changes, because really flux should be, the loss of material. And so you should see that in the change in those stocks over time, that the stock itself is not flux, but the change in stocks. It might be fun to look at that. So great stuff. Thanks.
Thanks so much.
Thanks again. Lauren has put a very interesting question in the chat. Lauren, would you like to ask directly, or would you just like me to read it out? And just unmute your microphone if you want to ask directly.
Sure, yeah. Thanks, Nicholas, for this great talk. I'm coming from Rutgers University. I'm a graduate student. I just thought this is a really great talk. I'm super interested in all the new applications of Biogeographical that have been coming out. This is a really interesting application. Yeah, so I was just wondering if there's any need to perform this kind of approach separately from the mesopelagic, like defining biogeographical regions for the mesopelagic itself. Because I know you're doing a lot of this definition for the epipelagic based off of chlorophyll and other parameters. But I was wondering if there's any need or interest to define the mesopelagic's own biogeographical regions based off of different parameters like nutrients, respiration, POC flux, etc. Or would we kind of expect the mesopelagic to kind of have those same biogeographical bounds and regions, being very tightly coupled with the epipelagic biogeographical regions that you've defined here?
So I think there would be, we haven't tried looking at it, but yeah, if we repeated the entire thing, only looking at the mesopelagic zone, only the region beneath the 0.1% per isolate, you would lose most of the source of variability. And so if you wanted to do that, then yeah, maybe using SVD might not be the most appropriate approach, or at least you would expect to have probably fewer biomes than we identified here. You would probably have the Arabian Sea stuff, where you have that really high concentration of backscatter or high backscatter values, the oxygen minimum zone, and maybe you have the high latitude time series where you had these larger particles. But then I think everything else might sort of be grouped together in a way that might not be totally informative. Again, we haven't tried looking at it, but that would just sort of be my my guess. Does that answer your question?
Yeah, I think that was great. Thank you so much.
Cool.
Thank you, Lauren. I think we've got time for another question. Dave Siegel.
Hi, Nicholas. Great talk. Thank you.
Thank you.
I have a technical question, probably because it's early here and I was making coffee as the talk started. How did you calculate transfer efficiency again? Because you had that interesting relationship with transfer efficiency versus the backscatter load, and I'm kind of curious how that relates to work that was done by Marseille and work by De Vries and Weber, et cetera, on what that relationship should be. Just curious.
Yeah. So it was really just the ratio of average backscatter within the productive zone, mesopelagic zone, divided by the average backscatter within the mesopelagic zone for each profile, and then averaged by season to try to account for any sort of time lags or things like that.
Okay.
And yeah, it was more or less consistent with I'm trying to remember the names escaping me, but it's consistent with other folks have found looking at float data. Sort of our additional contribution was that we were able to identify that this global inverse relationship is driven by what's happening at the high latitudes in summer or winter. In the winter, when you have these really high ratios, really low ratios, high ratios despite low concentrations of or low backscatter values within the productive zone. And then the other extreme, when you have these really high backscatter values and really low ratios.
Okay, that's interesting. Thank you. Can I just when you were doing the extrapolation, it seemed like you had an opportunity to test what Alan Longhurst did years ago, because you have all the global Argo data sets to look at it in a seasonal way, and it seems like it'll be interesting to see, do you get more fidelity in choosing these regions from remote sensing data products, or from just using what Alan did a million years ago?
Yeah, so we didn't go all the way back to the Longhurst provinces. We did compare our results, our predictions, to those that would be obtained using the variance bins used by Barenfeld et al., which was just based on chlorophyll concentrations. And what we found was that our results more or less aligned with the chlorophyll or satellite data results in cases where there were really high chlorophyll concentrations, in high latitudes, or where there were really low chlorophyll concentrations, in the gyres. Where it really added information was at the intermediate level. The original, based on variance bins, the intermediate biomes, or the intermediate variance bins, really cover vast expanses, and presumably vastly different expanses of the ocean. And the SVD results based on the Argo data were able to pull that apart a little bit more and differentiate, say, the equatorial stuff from the Mediterranean stuff, et cetera.
Can I ask the question a different way? Maybe I wasn't clear because I haven't had enough coffee.
Sorry.
Can you use physical oceanographic data alone to look at how those, does that separate the same provinces that you separated from the bio-optical data? Do you see?
Yes.
Because that's what Longhurst actually did.
Right. And so again, I think it provides a little more resolution. I can share one more figure. I hope this addresses the question. I'm not just sort of dancing around it. Just one sec. Let me share my screen again. But this shows a principal component analysis of the physical measurements from the floats. Just 1 second.
Yep.
So the physical measurements of the floats used to form this PCA, and then the individual points were colored by the biomes that we identified in our whole SVD clustering approach. And so you really get 3 clusters instead of 6, where the high latitude clusters are both differentiated based on these really high nitrate concentrations in the mixed layer depth, within the mixed layer depth, as shown here, and the higher backscatter values within the productive zone, while the Arabian Sea and equatorial biomes are both primarily differentiated by light levels at the nitrocline and the slope of the nitrocline at the nitrocline depth, as shown here. And finally, the Mediterranean and the central gyres are primarily differentiated by surface temperatures and salinity. So you can reproduce some of it, but I think the bio-optical data and the results that we obtained with our approach provide a little higher resolution than physical parameters would have alone.
Okay, thank you very much. I think we need to move along. So I'm sure Nick would be delighted to get any other questions from people if you'd like to email him.